And dear students, so in this presentation, I'm going to explain about immunity and the definition, classification, the general principles of various natural immunity, in particular phagocytosis mechanism, and what is acquired immunity, and the difference between active and passive immunity. So what is immunity or immunology? Immunology is a broad branch of biomedical science that covers the study of all aspects of the immune system in all organisms. Immune system is a system of biological structures and processes within an organism that protects against disease. The organs of immune system. So the immune system is divided into two types: primary lymphoid organ and secondary lymphoid organ. So what is this primary lymphoid organ? So where the primary lymphoid organs are thymus and bone marrow, and this is a place where the lymphocyte takes place of a maturation. And secondary lymphoid organ is the one which traps antigen and provides sites for maturation of lymphocyte, and it interact with the antigen. It includes lymph nodes, spleen, mucosal associated lymphoid tissue called MALT. And so many cells are involving in the immunity or immunological mechanism. So here I am briefly explaining about the cells which is responsible or based on our syllabus. And the blood cells is also having some contribution in the immunological function, including RBC, WBC, and platelets. So main the lymphocyte it constitute 20 to 40% of the body's white blood cells, and 99% of the cells in the limb. And these lymphocytes continually circulate in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organ. Thereby, it integrates the immune system to a high degree. And the lymphocytes can be broadly subdivided into three populations, mainly B cells and T cells. And also it is called natural killer cells on the basis of function and cell membrane components. This B cell and T cell is mainly involved in the humoral and cell mediated immunity. If the antigen possesses some antigenic site. So there are some antigens which doesn't contain any the antigens. There the natural killer cells it is having the major mechanism. And natural killer cells are the large granular lymphocyte that do not express the set of surface markers as like in the B cell and or T cells. What is this B cell and T cell? I'll explain you in detail in the next slides. So B lymphocyte is the one already we discussed. The B lymphocyte is derived, or first it was identified in the birds. And in birds, there is a place called bursa fabricius, and that is the place the B cell is getting matured. In the same way, in humans or in the mice, these B cells are getting matured in the bone marrow. So the B it corresponds three things you can take it. Either it may be derived from the bird, or it may be observed in the bursa fabricius, or it may be observed in the bone marrow. It is produced and matured in the bone marrow. And the mature B cells are definitively distinguished from the other lymphocytes by their synthesis and display of the membrane-bound immunoglobulin antibody molecules, which serves as a receptor for the antigen. The interaction between antigen and the membrane-bound antibody on the mature male B cells, as well as the interaction with the T cells and the macrophages, selectively induces the activation and differentiation of the B cell clones of the corresponding specificity. So in the next slide, I'll explain you completely in detail how this B cells it produce the memory cells for protection of antibody, as well as T cells in what way it acts. And in this process, the B cells divides repeatedly and it differentiates over a 4 to 5 day period, and it generating the population of plasma cells as well as the memory cells. The plasma cells is the responsible one. It is producing what type of an antibody. And the plasma cells which have lower levels of membrane bound antibody than B cells, it synthesizes and secretes the antibody. So this is what the structure of B cell, how it interacts with the antigen and antibody. So this is the mature B cell. It is derived from the bone marrow, which is containing this antibody. When the antigen it enters into the body, and this antigen it binds with the B cell. And during this 4 days and 5 days period, within this B cell, it recognizes what type of antigen, whether memory cells is it required to produce or not produce. And this B cells it's mainly react with if the antigen it is soluble in nature. So what will happen that antigen, it interacts with this antibody. So this B cells, it is having the power, it is having the surface marker of antibody. So the antigens which will bind into this antibody, and then it will divide it into this 4 days or 5 days period, it will be produced into two different cells. One it is called as the effector cell, or otherwise it is called as the plasma cell. This plasma cell is responsible for producing what type of antibody based on the antigenic site. So this antigen, each antigen, it is having its own specific epitope. Epitope, I'll explain you during antigen. So based on the type of epitope, this plasma cell will be producing specific antibody, and that antibody, it will react with the antigen, it will be neutralized. Whereas the memory cells, it will be produced in a type of case or type of infection. So the body decides for what cases memory cells it should be produced, and a few cases memory cells will be there, and few cases memory cells it won't be there. But still the mechanism we are not able to understand how the body decides. There are few cases why memory cells are produced, and there are few cases why memory cells are not produced. But most of the cases, wherever the memory cells are produced, those cases, if you see, all are the detrimental. So now we'll see what is a T lymphocyte. The T lymphocytes derive their name from the site of maturation called thymus. Already we discussed this one T cell. The second one, the like B lymphocyte, these cells also have membrane receptor for antigen. Although the antigen binding T cell receptor is structurally distinct from the immunoglobulin. It does share some common structural feature with the immunoglobulin molecule, but most notably in the structure of its antigen binding site. Unlike the membrane-bound antibody on B cells, the T cell receptor does not recognize the free antigen, whereas the T cell receptor, it recognizes only when the antigen that is bound to the particular classes of a self molecule. So that is why B cells are responsible mostly for a humoral immunity, and the T cells are responsible for cell-mediated immunity. And most of the T cells, it recognizes antigen only when it is bound to a self molecule which is encoded by the genes within the major histocompatibility complex. So what is this major histocompatibility complex? In the next presentation, I will explain you. And thus, the fundamental difference between the humoral and cell-mediated immunity is the branches of the immune system is that the B cell is capable of binding the soluble antigen, whereas the T cell is the one which will be binding only to the antigen which is displayed on the self molecule. Assume an antigen it enters into the cell, and then cell it will be presenting that particular antigen that is called as the antigen presenting cell, called as APC. And to be recognized by most of the T cell, this antigen must be displayed together with the MHC molecule called a major histocompatibility complex molecule on the surface of antigen presenting cells, or the virus infected cells, or sometimes it may be cancer cells, or it may be grafts. What is this mechanism? I'll explain you in detail during this phagocytosis mechanism. What is this natural killer cells? There is one more important part. So assume if an antigen, it is containing some epitope, our body it recognizes that is an antigen, and immediately that will be treated through neutralization reaction by producing an enough amount of antibody. But there are some pathogens, it doesn't contain any antigenic factor. It will enter and it will simply stay in our body. So those places, how this natural killer cells it acts or it produces, that you have to understand well. The natural killer cell was first described in 1976 when it was shown that the body contains a small population of large granular lymphocytes that display cytotoxic activity against a wide range of the tumor cells in the absence of any previous immunization or with the tumor cells. The natural killer cells were subsequently shown to play an important role in the host defense mechanism, both against tumor cells as well as the cells infected with some, but not all the viruses. And these cells, which constitute around 5 to 10% of the lymphocytes in the human peripheral blood and do not express the membrane molecule and receptor, that is the difference between T cell, B cell, as well as in the natural killer cell. Both T cell and B cell, which is having some surface molecule to distinguish the antibody, but this natural killer cell doesn't contain that receptors. And immunity, it is divided into two types, majorly innate immunity and acquired immunity. So innate immunity is the one which is non-specific and the other one is specific. And acquired immunity, it is divided into two types. One, it is active immunity and passive immunity. Again, this active immunity, it is divided into two types. One, it is naturally active immunity and artificial active immunity. Naturally active immunity is the one where a real infection and it causes and our own body it produces a memory cells like a chicken pox or something. When diseases happen and the antibody and memory cells will be produced and it will be long lasting. And artificial is the one without any infection. We are removing the antigenic factor and only with the immunogenic factor, we are introducing vaccines and we are making resistance against the disease. And this is called as the artificial active immunity. And here we are dependent on our own immune system, but the vaccines are introduced by either it may be killed or live or tetanus toxide. And passive immunity is the one, and it is produced by either natural, either through mother. Okay, mother is receiving the antibody, and that antibody it crosses into the placenta and to the fetus, and that is called as the passive immunity. And one more, it is artificial. Directly, we are injecting into an antibody. So this is what the passive immunity divided into naturally acquired passive immunity and artificially acquired passive. And again, this classification further is divided into two types in a complete detail. So this immunity, an innate or acquired immunity. So innate is the one, the first one is skin. Our skin that prevents the entry of most of the pathogens and microbes. And sometimes when you have minor cuts and cracks or some cuts are happening, through that the microbes are entered into the skin. And the skin secretions are the sweat, and that it always eliminates the microorganism, or the conditions are the salt what is present in the sweat, or the mucus, and that it elevates or it removes the microorganism from the surface, even tears and mucus and the nose. So those things are it removes the microorganism. And the phagocytosis, it's one important and a very interesting mechanism. I'll explain you in the next slide. And there is one more system, it is called REC, reticuloendothelial cell. And the inflammation, during inflammation, you can see that site, it will become reddening and that causes histamine. That chemical, it induces or it wanders the phagocyte, which is roaming around, and that phagocyte it is having a major mechanism in the inflammatory site and how it is directs the antigenic cell or antibody cells to act for that particular site. And natural killer cell, of course, it doesn't contain any specific antibody determinant, but it is having a strong reaction with an antigen. And of course, interferon for against viral and complement fixation system. So this will be a complement system. I'll explain you during antigen and antibody reaction. As like an acquired immunity, it may be a specific or non-specific. It is divided into two types, humoral and cell-mediated immunity. If the antigens are soluble in nature, so here the B cells which will be responsible, and it will be producing two types as we discussed in the previous mechanism. It will be producing a memory cells as well as the plasma cell. This plasma cell is one which is responsible for producing different types of antibody. So what type of infection and what kind of antibody, that is plasma cell, it decides during that 4-day or 5-day period of differentiation. And a few cases memory cells will be produced, but not all the cases. In this case, the antigen, it should be soluble in nature, whereas cell-mediated immunity is the one where the antigen it enters into the cell, and where the cell mediated immunity it will happen place. And once the cell it enters into the cell, and where we have a different mechanism like T helper cells and T killer cells and T suppressor cells, as well as like in the B cell, here also T memory cells will be produced, and that is responsible for the long lasting immunity. This is what about the T cell mechanism. And now the innate immunity as a system is what way we are born with, and it is non-specific, where all antigens are attacked pretty much equally. It is genetically based on, and we have that we are passed to our own offspring. And surface barriers are some mucosal immunity. It is specific for some individuals or some herd. And the surface barriers are mucosal immunity. The first and most important barrier is the skin. The skin cannot be penetrated by most of the organism unless it is having some openings such as nick, scratch, or cut, or else no microbes it will enter through this skin. And mechanically, pathogens are expelled from the lungs by the ciliary action as the tiny hairs which move upward motion. And coughing and sneezing, it abruptly ejects the microorganism. Sometimes, when we cough, whenever a pathogen or dust particulate enters, immediately we cough, and by the time, because of the forced action, the microbes or the dust particle or anything it will forcefully it will ejected from the entry. And non-living things from the respiratory system. And the flushing action of the tears, of course, we discussed already tears which is containing lysozyme, it also having its killing ability, as well as it is having that flushing action. When the tears are secreted, the microbes what it is attached on the eyes, it will be getting removed from the place. And saliva, it is also containing lysozyme, and it removes the microorganism or it destroys. And the urine, it also forces out the pathogens and as does not having a slogging of the skin. And the sticky mucus in the respiratory and gastrointestinal tract, it also traps traps many microorganisms, so that it won't allow the microbes to enter into our own system. And the acidic pH of the skin, it of skin secretions of inhibits most of the bacterial growth, because bacteria, as you discussed, it is most of the neutrophils or mesophile. It will be of the neutral pH only it will grow. When it goes for acidic pH, that bacteria it won't happen or it won't grow. And as we discussed in the previous slide, saliva, tears, and nasal secretions, and even the perspirations, if you see the major composition of the lysozyme, yes, in fact, Alexander Fleming was worked on this lysozyme initially to make it to concentrate and to treat an infection. But later stages, he found penicillin. But initial his work, it was completely depends on this lysozyme. And an enzyme majorly destroys the gram-positive bacteria. And of course, vaginal secretions are also slightly acidic, especially during the onset of menses. And that is the time period its most common infection will happen. That time, because of this, the acidic pH, and no infection will happen. And in male, spermine and zinc in the semen, it also destroys the pathogen. The sperms don't get infected with the pathogen. It's a natural protecting mechanism. And lactoperoxidase, it's a powerful enzyme that is found in the mother's milk. And during 2 to 3 months, okay, that innate immunity or during those period. And this lactoperoxidase is a powerful enzyme, and even the newborns, they don't have a matured immune system, and there the lactoperoxidase and lysozyme, it takes the responsibility of preventing the infection in the neonates. And the stomach, which is containing hydrochloric acid, it destroys most of the microorganism, and the stomach can even destroy the drugs and other chemicals apart from the pathogens or microbes. And phagocyte. This is what one repeated 5-mark question is repeatedly asked in the university examination. So to understand this phagocytosis only, whatever I given you as what is B lymphocyte and T cells. And what is this phagocyte? Okay, phagocyte is a cell that attracts by chemotaxis. Chemo, it is a chemical, taxis is a movement. A phagocyte, it will be around, in and around, it will move around in the blood circulation. Whenever there is a chemical is available to attract this phagocyte, there the phagocyte it goes and takes the action. And what will happen this phagocytosis mechanism? First it will go at the and then engulf and ingest the foreign bodies, which doesn't have any antigenic stimuli, or when the body is not recognized or not treated by either B cells or T cells. Those cases, this phagocyte is having the major functionary mechanism. And the pro-monocyte are made in the bone marrow, after which they are released into the blood. And it is called as circulating monocyte, which is eventually matured into the macrophages. What is this macrophages? Otherwise, you can call it as the big eaters. You don't require any antigenic site, or you don't have any specificity. Whatever the bacteria it comes, and this macrophages, it goes and observe that bacteria, and then it treats inside, and then it gives some kind of the coating substance on the pathogen. And later when it releases, the antibody system or our own immune system easily it recognizes those pathogen, and it will be treated nicely. So that is why this phagocytosis mechanism is one of the biggest immune system, or it's a repeated 5-mark question, because of its mechanism, or the interesting thing which is non-specificity, or how it destroys the pathogen. And these macrophages are two types of phagocytes are there. One, it is a fixed phagocyte. The other one, it is the wandering. Wandering is the one that moves somewhere without any particular purpose. Whereas B lymphocyte and T lymphocyte only with it moves to a specific target with a specific direction. But this fixed one and wandering one, it is in and around the body. It just moves around, and wherever it is required, immediately this phagocyte it acts and it produces phagocytosis mechanism. This is what the clinical or the microscopic image of phagocytosis, how it takes and how it destroys the pathogen, and it releases it to the environment within the phagocytosis. And some macrophages are concentrated in the lungs, liver, in particular in Kupffer cells, and where it is the entry of most of the pathogens, and the lining of lymph nodes and spleen, brine microglia, kidney mesangial cells, synovial A cells, and the osteoclast. And they are long living and depend on the mitochondria. This is our long living, and it is depends for mitochondria for its energy and are best attacking even the dead cells and the pathogens capable of living within the cells. So see, as we discussed, both B cells and T cells, if the antigen, it should be alive and it should have antigenic site as well as an immunogenic site. Assume the pathogen is killed, doesn't contain any immunogenic factor or antigenic factor, how those pathogens, dead cells within our body, how it is getting removed and cleared in sense that is through a phagocytosis mechanism. Because of that, this phagocytosis is very interesting mechanism, and it is an important immune function also. And these pathogens, it's capable of living within the cells. Okay, even the dead, and it will be survive or it will be available within the cells. And once macrophages, it called phagocytosis cell, it places some of its own protein called epitopes. This epitope is the one which is responsible for attraction of the antibody. Some antigens, when it enters, it itself, it is containing an antigen, so epitope, so that what will happen, the antibody immediately it will recognize and it will treat it immediately. Assume a pathogen it enters which doesn't contain any epitope, no immune function, it will act on the pathogen or maybe on the particle. That is why to treat, to get a good reaction, a good immunogenic reaction, the antigen should contain an epitope. So the antigen or pathogen which is entered into our own body, which doesn't contain any epitope, this phagocyte, it helps in the, first what it happens, the phagocyte, it absorbs the particular cell and it secretes its own protein, and it make in that particular pathogen, it produces that epitope. Later, when it is releases from the phagocyte, what will happen? Our antibody, it will easily, it will recognize the pathogen and it will destroy. So that is why phagocytosis mechanism is very interesting, and it kills even an antigen or the pathogen doesn't have any epitopes on its region. There is one example, some examples, no, simply the pathogen, it will enter and it will stay in our body. When there is no antigenic site or epitopes are not there, our antibody, it won't recognize. So it will be very harmful for us. But luckily, this phagocytosis mechanism, it helps us to treat or to neutralize those pathogens, including our dead cells. And on its surface, much like fighter plane, it displaying its hits. So simply, no, when you take the hits, fighting plane, if you see, there is a red color laser pointer will be there, and so that the point where you have to target it, that you can easily identify, and that is the exact mechanism of this phagocytosis. I don't think more than all these theory, whether you understood or not understood, maybe the next slide, the image what you are seeing, and that you can easily understand what is happening in the phagocytosis mechanism. And before going to that next slide, the image or the mechanism of phagocytosis, and little more theoretical aspects of this phagocytosis is, and the surface marker which serves an alarm to another immune cells, either it may be B cells or it may be called as T cells, and all cells performing this function, it is called as the antigen presenting cell. This is what APC I just explained you in the previous slide of the classification of immunity. And the non-fixed or wandering macrophages, it roam the blood vessels and can even leave them to go on an infection site where they destroy the dead tissue as well as and the pathogens. Immigration by squeezing through the capillary walls to the tissue is called as the diapetesis. Okay, it is the name of the mechanism or how the macrophages, it attracts or goes to that particular places, or otherwise you can call this extravasation. The presence of histamine at the infection site, it attracts these kind of wandering cells to their source. And that is why it is called as the chemotaxis. In the introduction slide of phagocyte, I told you it is macrophages or it's a chemotaxis. What is that chemotaxis in sense? The histamine, what is secreted in the infection site, where this diabetes or extravasation of the macrophage will happen, and it will go and accumulate in that particular place, and it takes or it absorbs or these big eaters, it eats the pathogen and it coats with some specific protein called surface marker, later will be released. And with the surface marker of the epitope, and it helps us to make this organism, it will easily treated by our own immune cells like antigen or maybe the antibody of B cells or T cell. And this is image, it represents this macrophages. It will be available through this wandering macrophages, will be available in the peripheral blood system. Assume this is the infection site, that blue color one. When the bacteria it infects here, suddenly the histamine which secretes, and that histamine it attracts the phagocyte where it is the target, where this phagocyte it goes and it absorbs this bacteria which doesn't contain any epitope in its region. Now this big eater, what it takes up all these bacteria and then it absorbs the bacteria, and inside it is processing. By the time when the bacteria it releases from the phagocyte, it is containing some amount of epitope. Later, the B cells or T cell, it comes into an action and then it destroys the bacteria, pathogen, dead cells, any organ or any foreign bodies which doesn't contain any epitopes. And there, this macrophages will have the major mechanism of treating, or it is internalization of this phagocyte, and it causes the neutralization. And this is what the exact or complete the viral mechanism which we are observed, or a microscopic observation in electron microscope. Assume the pathogen it enters into a particular body. This macrophage it goes and it absorbs the bacteria. And by the time after a few days, after a few hours, when it will be released from the phagocyte, this pathogen will be containing some epitope. And that epitope is the identification target for the pathogen, our own antibody, and immediately that later stages, this pathogen will be treated. So why this phagocytosis we are targeting very important in sense. Two reasons. One, it is the Mycobacterium tuberculosis, even it is getting internalized by the phagocytosis, the phagocyte cell, it cannot do anything. After Mycobacterium tuberculosis, when it there in the inside internalization, when it releases without getting affected, or it doesn't takes up any of the epitopes from the phagocyte, and it will come out as such as an inert Mycobacterium tuberculosis. So that's why Mycobacterium tuberculosis, it won't be treated through a phagocytosis mechanism. It will be survive years and years. Whereas a Staph, E. coli, or any other bacteria, when doesn't contain any epitopes, when it internalized by the phagocytosis, that will be easily treated through either B cells or T cell mediated immunity. But Mycobacterium is one exception. And today, in currently the technology, this macrophages are macrophage or phagocytosis mechanism, even we are treating it for treating the cancer treatment, where we know that already we say the macrophage is it is a chemotaxis, where some chemicals are available. This macrophages, it can be easily attracted. So in today's cancer treatment, what we are doing is we are targeting the cell which will be affected by the cancer. So you can add some epitope to the particular site. So the macrophage will be assumed that is a antigen, or it is a dead cell, or it causes some pathogen. It will absorb that own particular dead cell or the cancerous cell. Later will be released with epitope. So our own antibody will be treated. This is one today's treatment which we are following in the chemotherapy or in the cancer treatment. So that is why this macrophage or phagocytosis is very, very interesting and important thing in the immunological system. And the next one is cell-mediated immunity, and macrophages engulf the antigen and process them internally, then displace the parts of them on their surface together with some of their own protein. That own protein, what is that own protein? It is called as cluster differentiation, or what it is called as the CD. And I'll explain you. This sensitizes the T cell to recognize these antigen. As initially, when the pathogen doesn't contain any antigenic site, so that is why our body cells it is not able to recognize, or it is not able to treat it. But once it is phagocytosis is over, by the time what will happen, when it releases from the phagocytic cell, it will be containing a specific target or specific protein. That the protein name or the specific target region, it is called as the cluster differentiation, CD cells. Okay, this CD, it is recognized by T cells as well as B cells, and later that pathogen will be treated nicely and without harming any of our own body function. And here CD, it stands as nothing but it is the cluster differentiation. And there are more than 160 cluster differentiation cells are identified so far, each of which is a different chemical molecule. Even this CD, CD8 plus if it is present in the phagocytosis, and that is treated only through a T cell. And there are some cluster differentiation if it is there, and that will be recognized by the B cell. Again, you see the mechanism, the wonder of a body, and the phagocyte itself, it will decide how it can be treated. Either it should through a T cell, or it should through a B cell. If it is need to be treated through a T cell, where it is coated with the CD8 plus, it is called CD8 positive. And every T cells and B cells, it is having its own CD cluster differentiation molecule. For a B cell cluster differentiation, assume the pathogen is coated with the CD21, that will be treated through a B cell as like CD35, CD40, CD45, in addition to other non-CD molecules. And it's not necessary it should be CD molecules. And apart from CD molecules, there are some other chemicals also will be coated on the phagocyte so that the phagocyte easily it will recognize. But once it is coated with the CD21, and that will be treated only through a B cells, not by the T cells. So what is in the case of T cells, how it should be recognized in sense, a T cells which is having a CD2 or CD3 or CD4, CD28 or CD45 or and other non-CD molecules on their surface. These cluster differentiation molecules are there in the pathogen after internalization when it is exit from the phagocyte, that will be treated only through a T cells. And apart from that, the general identification or general information about the T cells, the T cells are called cytotoxic or killer T cell, which is called CD8 plus, CD8 positive, and it will do their work by releasing lymphotoxins. Okay, so within the lymphocytes, it will release some toxins, and so that it causes the cell lysis. And helper T cell, that is called CD4 plus, it serves as a manager. It is directing the immune response. Okay, unless only CD8 plus, it won't be functioning. CD4 plus also it is helping in directing the T cells to the internalized cells. And it secretes the chemicals, it's called the lymphokines, that it stimulate the cytotoxic T cells as well as the B cells to grow and divide and attract the neutrophils and enhance the ability of the macrophages to engulf and destroy the microbes. And the memory T cells are programmed and recognized and to respond to a pathogen once it has been invaded in and it is repelled. And finally, the acquired immunity. So here I've got some general information. The resistance that a human acquires during the life is known as acquired immunity. These are two types, as we discussed in the previous classification, active and passive immunity. Active is the one, the resistance developed by an individual as a result of antigenic stimulus. And passive is the one directly we are receiving an antibody. And active immunity is the one, it involves active functioning of the host immune apparatus, synthesis of antibodies, and production of immunologically active cells, and it develops either humoral or cellular immunity. And here there is some latent period. It requires time for activation, and there will be negative phase also, a time period in which there is a reduced measurable immunity where antigen combines with the pre-existing antibody and lower its circulation. And the secondary response, if an individual previously exposed to same antigen, again will respond quickly and abundantly. First time exposure only, it will take some time to produce an antibody, but once first time exposure and memory cells are there, second time exposure, even without any symptoms, that antigens will be neutralized. And it also associated with immunological memory cells, but everywhere the memory cells, it won't be produced. And it will be more effective and it will be better protection than the passive immunity. So that is why always preference is always, it goes for active immunity. And passive immunity is the one that is for a short duration. Okay. And clinical or inapparent infection by a microbe, and immunity is long lasting. So here, what is the active immunity? The immunity is a long lasting, but it varies on types of pathogen. And large majority of adult, we are getting an active immunity against the poliovirus. And there are sometimes lifelong active immunity we are getting specifically for chickenpox, where memory cells will be produced once it occurred, and after that it won't occur. And with the exception of influenza and common cold, you may ask, common cold, why we are periodically we are getting instance. Every time the virus, it changes its own antigenic variation. So that is why even in this case, a common cold virus, the plasma cells or body, it won't produce any memory cells. So that is why I wonder, the important thing what we can wonder is how the body recognizes why few cases memory cells are produced, why few cases memory cells are not produced. So those cases memory cells are not produced, that it won't be a detrimental, it will be of just normal infection and it will causes 1 or 2 days of infection. And influenza, it may reoccur after a few months or a year due to lack of immunizing capacity of the virus. Sometimes the virus, it won't produces the immunity, immunization or memory cells producing effect. And common cold is having same clinical feature caused by other viruses. And immunity following by bacterial infection is less permanent than viruses. And active immunity called artificial. So here, resistance induced by the vaccines, it may be live, killed, or attenuated bacteria attenuated strains. It may be either bacteria or through a viral vaccine. And here, one more important term you have to understand, it is booster dose. And sometimes, on the first dose of vaccine, it won't be produced a sufficient amount of memory cells, or and the effect of antibody, it won't be long lasting. But when you receive the second dose, the immune system will recognize, okay, this infection, it will happen again and again. Then the function, it will be long lasting. So that is why there are few cases booster dose is required. There are few cases a booster dose is not required. But again, it's a major confusion, or the mechanism we could not understand why there are few cases booster dose is required. But there are few cases even in the first dose itself, we are producing a strong and permanent memory cells. So again, if you see the disease difference, you can understand the booster dose. A few cases we require and few cases we don't require at all. And so examples of a few active immunity. Here, it may be in the form of live vaccines, for example, BCG, Bacillus, Calmette, and urine, and the killed vaccines, example, typhoid vaccine, and bacterial products. And sometimes, the products of bacteria we are taken. And one example, it is tetanus toxide, TT. And active immunity through live vaccines, it initiates the infection without causing any injury or disease, and it parallels with the natural but lower grade, which is administered orally, Sabin type polio, the one which we are having. There are two types of polio vaccines. One it is Sabin type, and the other one it is a SAC type. Sabin is the one it is alive, and the SAC type is the one it is a dead bacteria. And parenterally, what we are administering, it is called measles. And the active immunity called artificial, it's called killed vaccines. Example, it is less immunogenic compared to live. Always live is a better immunogen, and it will be producing even killed vaccines. We are producing long-lasting memory, and protection lasts for short period. They have to be supplemented by the booster doses. Most of the killed vaccines, if you see, booster doses are compulsory. And the parenteral administration, the humoral antibody response improved by the addition of adjuvant. Apart from antigen or bacterial byproducts are killed or attenuated, there's some additional of some substances. For example, aluminum, when you added along with the toxide, it will be increasing the memory cells producing ability. That substance, what it is called as the adjuvant. Adjuvant, along with the antigenic substance or immunogens, we are adding some substance to produce the enhanced immunological effect, either through memory cells or for the production of antibody. That agent, it is called as the adjuvant. And passive immunity, as we discussed in the previous case, directly we are administering antibody, but the administered antibody, even it is getting transferred to the baby during the pregnancy condition. If the mother is receiving antibody, that will be transmitted to the placenta in the humans. But very interesting thing here, if you see, you administer 1 milligram of an antibody, for example, immunoglobulin A, and that is present more amount in the neonat than in the human. So the maximum amount of protection, it is given to the neonates. See the system of nature system. Whatever the antibody receives by the mother and during infection, more amount of antibody will be entered into the fetus than into the mother. See the mechanism, it is even acting against concentration gradient. Generally, a cell or a concentration or the cells will be go for equal concentration as per our osmosis principle. But in this case, during infection, if a mother is receives any antibody, and that antibody, more amount of antibodies will be present in the neonate than in the human mother. And human fetus ability to synthesize immunoglobulin M from 20th week of gestation is reported, and some part of immunological independence it develops only after 3 months period. Until then, the maternal antibody, it gives the passive immunity. Generally, neonates are not exposed to other place. That's why neonates are well protected without moving from one place to another place. By the time the immune system, it is immunological system will get ready, and so that they are able to protect themselves. Their defense system will be developed. So that is why neonates are more prone to an infection. We have to be protected as much as possible. And passive immunity, natural, it transports the antibodies across the placenta. Is an active process, and concentration in the fetal blood is higher than the mother. So as we explained in the previous slide, infection in children is common after the age of 3 months and than in the neonates. An active immunization of mother during pregnancy, it improves the quality of passive immunity in the infants. That is one more important point. And tetanus toxide is recommended during pregnancy, and that will protect the newborn more than in the who is receiving the vaccines directly them. From the mother when they received, and then the antibody development is more in the neonates. And passive immunity artificial. The resistance is passively transferred to a recipient by the administration of an antibody. This is by hyperimmune serum. So we are injecting immune. So already we discussed the serum immune serum preparation. We inject antigen and then we collect the antibody and then we are administering into an individual. There is one more sera we call it as convalescent sera. What is called as convalescent sera? I am having an infection, and if you collect my serum, I am having that antibody for the against for the infection which I occurred. That serum treatment, it is called as the convalescent sera. This is what we discussed during the COVID condition. People, they were expected this convalescent sera to give for treating the individual who is not able to recover from the COVID, and pooled human gamma globulin. And this is one that is a we can collect it from each individual, and later that it can be treated or it can be given to an individual who is infected. And the final one, the differences between active and passive immunity. This is what one repeated 5-mark question in the university. Apart from phagocytosis, the second important 5-mark what you can expect is the active and passive immunity. So what is active immunity? Produced actively by the host immune system by the infection or through vaccines, and it is induced by infection or by immunogen. Immunogen as an agent which is containing an antigenic factor, and it is effective protection for long lasting, and immunity is effective only after a lag period. Initially, we need to spend 1 or 2 days. After that, only we are able to get that protection. And booster dose is effective, and negative phase it may occur, and it is not applicable if the patient is immunodeficient, and it is suitable for naturally active tuberculosis and artificially active for BCG vaccine. Passive immunity is exactly opposite what we discussed for active immunity. It is received passively, ready-made antibody we are administering, and here less effective, but we have only advantages. Here immediate immunity is there, but in this case, no memory cells will be produced in passive immunity, and the subsequent dose is less effective, and there is no negative phase in the passive immunity. So that is why in emergency cases, we can't go with the active immunity. We have to go with the passive immunity. And it is applicable in immunodeficient individuals. Example, natural passive, resistance passively transferred from mother to baby, or artificial passive administration of the collected antibody either through immunoserum or the convalescent serum. And nowadays we are following a combined immunization is the one